Hey guys, today we'll study something about Coombs test. Coombs test is of two types, direct Coombs test and indirect Coombs test. Let's just find out what is this test all about. The two types of test, where are they used and what is its importance? There is one thing similar between these two types of tests, which is the Coombs reagent. Let's find out what is the Coombs reagent and how is it produced. So basically, Coombs reagent is nothing but the anti-human globulins, which is produced inside an animal. Let's find out how is it produced. We take a blood sample from a person who is suffering from immunohemolytic anemia or any kind of disorder in which the autoantibodies are produced against the antigens on the RBC. These antibodies or the blood sample is first processed and it is washed. The sample contains autoantibodies. It is then injected into an animal, maybe a rat or maybe a hamster. The injected sample stimulates the animal to produce antibodies against these human antibodies. We take out the plasma from the animal which contains these antibodies, which is called as the anti-human antibodies. They are also called as the anti-autoantibodies, or rather the anti-antibodies. These antibodies are the antibodies which are produced against the human antibodies, and that's what Coombs reagent is called. Coombs reagent is nothing but it is the plasma which contains the anti-human globulins or the anti-human antibodies. Let's study direct Coombs test in detail. How is it done and where is it done? Now in direct Coombs test, we are using the autoantibodies which is already present inside a person or a patient who is suffering from immunohemolytic anemia. We take the blood sample from the person and introduce into a test tube. The person who is suffering from such disease has the antibodies which is attached to the RBCs, whereas the normal RBCs do not have any kind of autoantibodies attached to it. We then add the Coombs reagent into both the samples, both the test tubes, to find out whether the coagulation occurs or not. The anti-human antiglobulins bind to the autoantibodies and they form a coagulation or a coagulum which is visible to the naked eyes. Whereas the normal blood does not coagulate because of the absence of any kind of autoantibodies. When the coagulation occurs, it forms a visible mass which is present at the bottom of the test tube. This is suggestive of a positive DCT, whereas in which the coagulation does not occur tells us about the negative test. Positive direct Coombs test indicates that the autoantibodies were present in the sample, which is suggestive of the fact that the person is suffering from any kind of autoimmune hemolytic disease. Now let's just talk about the indirect Coombs test and how is it different from the Coombs test. The indirect Coombs test is done in an Rh negative female who is having a baby who is Rh positive. This is done to prevent the Rh iso incompatibility or Rh isoimmunization. In this test, the mother's blood is taken and from the blood the plasma is extracted. Whether the plasma contains these antibodies or not shall be confirmed by the indirect Coombs test. Now, this is the basic difference between the direct and the indirect Coombs test. In the indirect Coombs test, we are adding just the antibodies which contains the plasma without the red cells, and we are adding the red cells externally which are Rh positive. After having added these two things in the test tube, we then add the Coombs reagent to it. The Coombs reagent contains the anti-human globulins which bind to the autoantibodies if present, and if not, they don't bind. Presence of coagulation indicates a positive indirect Coombs test, whereas no coagulation indicates a negative Coombs test, which means there is no autoantibodies present. In the indirect Coombs test, the ICT positive indicates that the Rh negative mother is already sensitized, whereas the ICT negative confirms that she is safe and not yet sensitized.